Picture this. You're staring at the most complex, heavily guarded vault in the entire universe. You know for a fact that the deepest secrets to human health and human disease are locked right inside there, but you simply don't have the key. For decades, biologists faced exactly this frustrating reality. Gene editing basically felt like working inside a pitch-black, locked room with incredibly clumsy tools. Sure, scientists could peer through the keyhole of human biology, they could occasionally, break a window to see what happened, but true, precise control? No way. That remained completely locked away in the dark. Welcome to this explainer on CRISPR, promise, proof, and ethics. Today, we're going to explore how a humble, completely natural bacterial defense mechanism somehow transformed into a Nobel Prize-winning revolution. And really, this isn't just a story about a cool scientific tool. This is the story of how humanity finally unlocked the very secrets of our biology, and why the absolute hardest questions we face right now are no longer just scientific ones. Part 1. The Unlocked Room. Why CRISPR changed everything. Okay, so think about that microscopic, utterly chaotic code of life. Suddenly, almost overnight, that dark room was flooded with light. What used to be entirely out of reach just became possible. The entire scientific process transformed. Experiments that were painfully slow became infinitely scalable, way faster, and remarkably cheaper. But honestly, the most crucial part, the realm of what was even thinkable fundamentally changed. The very ceiling of what scientists could imagine achieving shifted permanently upward. It was a total game changer. Part 2, a biological GPS system, search and target. Now, to truly grasp this, you kind of have to understand the sheer, staggering scale of DNA. Our genome is an enormous library. Finding a single disease-causing mutation is literally like hunting for one specific misspelled letter hidden within millions of towering books. Before CRISPR, trying to edit that felt like driving across the country using outdated paper maps. But CRISPR acts as a highly programmable shipping label. It's essentially a biological GPS app. The GPS sets the precise destination in that massive library. But, and this is a huge but, it doesn't actually move the car for you. It tells the cellular machinery exactly where to go without doing the heavy lifting of fixing the gene itself. And the absolute genius of this biological design lies in the modular shift. Think of it like a universal power outlet with interchangeable chargers. In the CRISPR system, one core mechanism stays exactly the same, while the targeting component, the guide, can be quickly swapped out for whatever genetic address you're trying to find. This incredible modularity turned what used to be a really difficult one-off parlor trick into a universal, infinitely reusable platform. Part 3. Editing is not repairing. The illusion of the typo fix. We constantly hear the phrase gene editing, right? And we naturally imagine just hitting backspace to fix a typo on a computer screen. Well, scratch that, because biology is incredibly messy. The process actually happens in 3 very distinct phases. First, there's targeting, getting to the right spot in the genome. Second, there's the editing itself, where CRISPR creates a physical break in the DNA strand. But it's the third step where that whole computer typo illusion totally breaks down, and that is repair. While scientists can perfectly control the targeting and the cut, this final, absolutely critical step is entirely at the mercy of the living cell's own unpredictable response. Which brings us to a really sobering biological reality. Precision is not the same thing as predictability. We have this jaw-dropping power to cut DNA exactly where we want it, but the cell, it might repair that break in a way we completely didn't expect. The human plan might be flawless on paper, but the living, breathing organism always has the final say. This creates a really fascinating divide right at the frontier of science. On one hand, testing gene functions became vastly easier. We can now alter a single biological ingredient just to see how the whole recipe changes. We're finally separating mere correlation from actual cause. But on the other hand, the genome still fiercely resists our total control. We are constantly battling against those unexpected cellular repairs and targets that are incredibly hard to reach. Because a gene isn't just a simple on-off switch, it's a deeply connected node in a vast, complex network. Part 4, the delivery gatekeeper, from lab to clinic. Okay, let's zoom out for a second. We're moving from the microscopic environment of a single cell to the vast, turbulent ecosystem of the human body. Because once a tool works perfectly in a nice, controlled petri dish, the defining question becomes, can we do this safely inside a living human? The body is not a clean laboratory. It's an active, highly defensive fortress. Scientists have to clear some intense biological hurdles. They have to physically reach the right tissue, evade an immune system that is literally designed to attack foreign invaders, and then ensure the edit is actually durable enough to last for the patient.
This creates two vastly different realities for clinical treatments. First, there's ex vivo delivery, which happens outside the body. Cells are physically removed, edited in a safe lab environment, heavily checked for accuracy, and then returned to the patient. And this is exactly where our early, incredible successes in blood disorders have happened. But then there's in vivo delivery. That means injecting the CRISPR cargo directly into the body, forcing it to navigate that chaotic environment completely on its own. It holds vastly broader potential for sure, but it is infinitely harder to target and monitor safely.
Part 5. Power meets responsibility, where science meets society. As we unlock all these monumental capabilities, we really have to shift our narrative. We need to move from asking what is merely scientifically possible to asking what is ethically justified, because a technology can be scientifically brilliant, yet remain ethically unstable. We have to learn to actively weigh CRISPR, not just admire it. We need to separate real clinical advances from overclaim. We must recognize the profound truth that just because an edit is possible absolutely does not mean it's appropriate. Public ethical debate isn't an obstacle to the science. It is the cornerstone of responsible science. Ultimately, the deepest question we face as a species isn't really about the microscopic mechanics of cutting DNA anymore. It's this. With the very code of life resting securely in our hands, what should we change? For whom? And under exactly what conditions?